Hello everyone, I'm Divya. I'm currently a PhD student in Dr. Corey Nislow's lab at the University of British Columbia. Today, I will be introducing you to my project, which is titled as a systematic strategy to link human genetic variations to drug response. I'm also presenting a detailed poster about it today, so please come and check it out. To me, it is interesting to hear that for any particular disease, every patient is administered the same drug in the same doses, even when each of them react in a different way to the drug. To date, clinicians still can't predict which patient should be prescribed certain medications and in what dosages, and if those patients will suffer any marginal or acute side effects. In my opinion, as a researcher, if I can apply pharmacogenomics to decipher what influences the effectiveness of drugs and make the results accessible to the clinics, doctors will be able to prescribe the best drug at the appropriate dosages to their patients. This whole narrative kind of forms the basis of my project, and the overall idea is to combine all the advancements in next generation sequencing with chemical biology to get an improved understanding of genomics and how it drives an individual's health and response to drugs. So, keeping all that in mind, the first question that I'm trying to answer is which genetic differences are responsible for variations in efficacy and toxicity of drugs? And following this, I would like to know how to assess the true functional impact, that is, the fitness and drug response of these clinically actionable variants. And lastly, if it is possible to use the results from AIM-1 and AIM-2 to make predictions on the impact of new variants in patients. But tackling such objectives is not easy. The main challenge here is the sheer number of variants between any two individuals. It's between 3 to 4 million. The solution to this problem that we have come up with is to devise an approach that can quantitate all variants by capturing drug response in the target gene, leading to their functional characterization in terms of drug resistance and sensitivity. So, this platform or strategy that I'm trying to develop and deploy is called variant technology or VTech. The idea is that for any drug target pair, VTech will be able to convert variants of unknown significance, those that affect phenotype, into actionable variants. Now, this figure shows what the VTech strategy involves and what will be implemented. It involves creation of a site-saturated variant library through mutagenesis, cloning of the library into a plasmid vector, variant expression and auxotrophic selection of the transformed yeast cells, because we use yeast in the lab, deep sequencing the samples extracted at different conditions, processing the next generation sequencing data for variant detection, validation of the variants associated with drug response, and finally, the functional characterization of actionable variants. Let's talk about the variant library design and synthesis. To test the VTech approach, the first drug that I decided to evaluate was methotrexate, which is frequently prescribed for cancer, rheumatoid arthritis, and severe cirrhosis. The target of methotrexate, dihydrofolate reductase, is well established, and its human ortholog, DHFR, is known to both complement the yeast homolog DFR1 and confer drug resistance when overexpressed. So after selecting the drug target pair, I wanted to design a supervariant library of DHFR. And like you see in the slide, DHFR has 187 amino acids. And for the design, each position of the amino acid was substituted with all possible amino acid variants to make a site-saturated or variant library design. We use non-synonymous and E-specific codons in the design, and all stop codons were eliminated from the library as well. Finally, I sent the modified plasmid and the completed library design to Twist Biosciences for production, as they have an optimized workflow that allows for a massive parallel oligonucleotide synthesis. They basically helped us build a high quality, precise, and uniform library, which was NGS validated at each amino acid position. This slide shows the overall workflow for the variant library synthesis. The first step was the variant library design, followed by gene synthesis, library production, onboarding that a newly built library on a customized plasmid, followed by NGS verification of each residue of DHFR. Now that I have this super precise variant library, the next steps in the project will be manipulation of the deployed variant libraries to haploid to check for variant survival and viability, followed by drug resistance and sensitivity screening assays, PCR amplification and library preparation, NGS, and data processing. And that will be followed by variant analysis, which will include different steps. Of trying to understand the fitness and drug response of each detected variant, their preferences at each residue site, and also we'll try to map the variants on the DHFR structure to understand the sequence structure function relationships. And all this will be followed by trying to develop a prediction framework to assess new variants in patients. And for the results, I expect to generate and deliver different rosters of variants ranked according to resistance, sensitivity, and time or dose dependent dynamic resistance to the clinics. And at this point, I would like to acknowledge my supervisor, Dr. Corey Neslo, for all his insights and support, and the entire lab committee and funding sources. Additionally, please feel free to come check out our lab website and contact me if you see something interesting or would like to connect. Thank you so much.